Okay, that's me. Hi everyone, my name is Charlie. I'm a postdoc working for Andrew Roger and Ed Susko out in Dalhousie, and today I'm going to be talking about some of the work we've been carrying out on complex models of protein evolution. So I just want to first define what I mean by site and branch heterogeneity. So site heterogeneity is the idea that different sites in your alignment are going to undergo different amino acid substitution processes. And so when you hear people talking about site heterogeneous models, generally what they are using are the two things I've labeled here in red. A substitution matrix like LG or WAG, which defines amino acid substitution for a single site, and then a profile mixture model such as C10 or C20, which in combination with this substitution matrix allows for substitution processes to vary across sites of an alignment. So branch heterogeneity then is the idea that the amino acid composition can vary across different branches of your tree of interest. There are models which do try to account for this variation across branches, but they tend to be more complex and parameter rich, and more importantly, they're a lot more computationally expensive than site heterogeneous models, so you don't see them being used as often. So why would you want to model site and branch heterogeneity together? Well, the example I'm going to give you here is from a paper by Foster and Hickey, published when I was about 6 years old. So what they did was they took a bunch of mitochondrial sequences from 8 Apistecon taxa, and they looked to see what heterogeneity across these sequences might have, what impact that might have on inferring a species tree from these sequences. So in the middle, I have a table which gives some information for the sequences that they sampled, and I've highlighted the information for honeybees and nematodes. The sequences that they sampled for this data set are particularly AT-rich, and so on the protein level, that means they're rich in thymine amino acids and very poor in GARP amino acids. So as regards to inferring a tree from this data set, on the top right is the correct biological consensus tree for these taxa, where nematodes branch at the base of the ecdysozoa clade. However, what generally happens is if you ask IQ-Tree or some other software to infer a tree from this data set, and you don't provide a model which accounts for branch heterogeneity, you'll probably get a tree that looks like something on the bottom right, where nematodes and honeybees branch together. So this is a classic case of mismodeling heterogeneity leading to long branch attraction artifacts. So another example of heterogeneity over a tree, which is going to be relevant to what I'm going to show you later on, is this question of the relationship between mitochondria and alpha proteobacteria. So whether mitochondria are sister to alpha proteobacteria, or whether they branch within alpha proteobacteria. And what complicates this, resolving this relationship, is that both mitochondria and several lineages within alpha proteobacteria have undergone convergent genome reduction and thus have very AT-rich genomes and very thymic-rich proteomes, whereas other alpha proteobacteria are more GC-rich and have more GARP-rich proteomes. So feel free to go and read the paper to get a more detailed explanation of what they did here, but basically what's relevant to my talk is that this paper implemented a new GF-mix model to support the placement of mitochondria outside of the alpha proteobacteria. So as a very quick and dirty and not particularly mathematical summary of what GF-mix does, this was developed by Ed Susko in Dalhousie, and what you do is your first step is you fit a site heterogeneous profile mixture model to your data set and tree of interest using IQ-Tree. And then in the second step, you take that data from IQ-Tree and you give it to the GF-mix software. And what GF-mix will do is for each branch of your input tree, it will add a new parameter which represents the ratio of GARP to thymic amino acids estimated at that branch. So in the graphical example I have here for taxa A, that has a ratio of 1.5, so there should be more GARPs than there are thymics. For the other example, taxa D, that has a ratio of 0.25, so there should be more thymics than there are GARPs. And what GF-mix will do is, for each branch of the tree, it will modify the underlying profile mixture model so that the frequencies of the model better reflect amino acid composition at that branch. Once it's done so for every branch of the tree, the likelihood is then re-estimated as normal. So some of the work that I'm going to be showing you today as part of a larger project has been testing and validating improvements we're making to the GF-mix model. So the main drawback of the original approach in that 2022 paper is that we can't do any reoptimization of the model parameters or branch lengths that are inferred or estimated by IQ-tree. We're stuck with them. So Ed has developed a new method which uses maximum likelihood first to estimate branch-specific composition parameters, but using that we're also able to re-estimate branch lengths and other parameters of the model such as weights and alpha. Because it's implemented in maximum likelihood, that means that for quite large and complex data sets, this might be quite slow. So what we're also looking at is an approximation of this new method, also developed by Ed, which doesn't do reoptimization of model parameters, but what it does try to do is it estimates branch-specific composition parameters that try to minimize the sum of squares difference between the observed frequencies in your data set and across your tree and the stationary frequencies implied by the underlying mixture model. So this may have some speed improvements over the full version. So as I'm testing this out, we're using data which has been simulated to exhibit a GARP Phylogenomics composition bias over the tree. So I've been simulating this data using AliSim, which is part of the IQ-tree software package. So some of the results that I'm going to be showing you are for 100 simulated replicates, which were simulated using a C20 profile mixture model for site heterogeneity. Then Min Bui's group, and particularly Nan, within Min's group out in Australia, who maintain IQ-tree, they were kind enough to give us some guidance for simulating or for constructing a custom pipeline to simulate branch heterogeneity across a tree, as this is not currently a built-in function of AliSim. So feel free to ask me about that later on. So just some information on what the replicates look like. So we simulated for 10,000 sites, no gaps in the alignments. We simulated using a 16 taxa tree, and then we downsampled these alignments to 4 taxa. And some of the questions that we wanted to ask with the simulated data were, how well do our various versions of GF-mix do at estimating composition, branch compositions, and branch lengths when we have already defined what the true value should be, and how well do they perform when we downsample taxa and thus have less phylogenetic information available. So this is just the trees here on the left is the simulating tree, so any branches that are in black should have equal amounts of GARPs to Phylogenomics, and any branches which are in blue should have more Phylogenomics than they have GARPs, and then there's the downsample tree on the right. So for estimating branch lengths, what we're comparing here is whether we don't do any optimization of the branch lengths using maximum likelihood versus if we do reoptimize branch lengths using maximum likelihood. And so I'm giving these values as mean squared errors. So the smaller the error, the better we're doing. And so hopefully what you can see from this table here for the 16 taxa trees at the top and the 4 taxa trees here at the bottom is that when we do reoptimize branch lengths, we're doing much better at getting closer to what the true branch lengths should be for the clades up on the top for the 16 taxa trees and for the terminal branches for the 4 taxa trees. The payoff for this is that we're not doing so well we're not showing any major improvements when it comes to estimating branch lengths at the internal branches. So there's a little bit of a trade-off there. So then for estimating GARP composition, GARP Phylogenomics composition across the trees, so I'm giving an example here from the 4 taxa trees. So this box plot is from 100 4 taxa replicates. So on the y-axis is the estimated GARP to Phylogenomics ratio, and then the x-axis is the 4 different node types in the 4 taxa trees. So our equal GARP Phylogenomics on the left, followed by our high Phylogenomics, then our internal and root nodes on the right. And so our equal GARP Phylogenomics, our internal nodes, and our root nodes should all have roughly equal amounts of GARPs and Phylogenomics present, whereas the high Phylogenomics nodes should have more Phylogenomics than they have GARPs. I've indicated those lines there on the plot. And so what you should hopefully see for our 3 methods, the old method in red, the approximate in green, and the new method in blue, is that our old methods are sort of either underestimating or overestimating the amounts of Phylogenomics that should be present at these nodes in the 4 taxa trees. However, when we move to our newer, more complex methods, we're seeing them getting closer to what the expected values should be. So we are seeing some improvements in estimating composition parameters, and this is also true for the 16 taxa trees. So finally, I just want to give you a quick example of how we're applying this to real data. So I'm going back to that Foster and Hickey paper from 1998, where they sampled a bunch of mitochondria sequences and looked to see what effect heterogeneity was having on tree inference. So we've got two hypotheses to test out here. The one on the left is the correct tree, where nematodes are at the base of Ecdysozoa, and the other one is the tree on the right, where honeybees and nematodes are each other's closest relatives. So the likelihoods I have here are starting from the top for a LG C20 plus gamma site heterogeneous model as taken from my Q tree, and then using that as a base, testing out what happens when we apply different versions of our GF mix model on top of that base model. And so hopefully what you can see is that for our base plus new GF mix model down at the bottom, we get just a little better likelihood for the correct tree over the incorrect tree. Really what it means is both trees are equally likely. So in an ideal world, we'd love to see, just get 100 likelihood unit difference. We're just smashing it, but we're not getting there yet, but we're moving in a positive direction. So we can look at varying what the input mixture models we use are and other things. So to conclude, our GF mix model tries to address compositional heterogeneity by making branch-specific modifications to site heterogeneous profile mixture models. Applying this to both simulated data and real data suggests that our newer, more complex versions of the GF mix model are better addressing heterogeneity artifacts. And I just want to say that this is not the only work we've been doing on this project. We've also been looking at extending the GF mix model to model other composition biases, not just GARP and Phyminx. So we were collaborators on a paper that was recently published looking at halophilia and archaea. That's in Nature Microbiology. It's very cool work. And we're also looking at ways of estimating composition from data directly and applying this to larger scale real data sets. And so with that, I will leave you with acknowledgements of my collaborators in Dalhousie and collaborating labs in France and Australia, our funders, and thank you for listening. Yeah.
Do you think that what you talked about is primarily for really deep divergences, or do you think that it's applicable in shallow divergences?
It might be applicable in shallow divergences if I guess you know that there's some specific compositional difference between the two. I can't think of any examples off the top of my head, maybe within yeasts or something like that. But yeah, I mean, it's definitely, it's applicable where you think that there's going to be compositional effects on the final tree. Yeah?
Can you talk a little bit about the computational time differences between the new approximate and the old?
Well, the old version, it's sort of using nonlinear equations and things to sort of get at the estimating parameters, so that's not particularly intensive. Really, with the other versions, it depends on how large your trees and your alignments are. I haven't really necessarily looked at how this scales, but it very much depends on the amount of taxa and the sort of length of the alignments you're looking at. Another minute or so. All right, thank you.
Okay, I'll get started. My name's Todd Jackman. I'm from Villanova University. I'm going to be talking about rapidly evolving long exons today. So let's just see. Okay, there's really three things that I want to discuss today, and this stems from a 2020 paper that Ben Karen, Tony Gamble, and myself published. And since then, we've added 100 loci and maybe about 300,000 bases. So we've got about 1 million bases that we're looking at that we consider to be long exons. It's really a comprehensive set of rapidly evolving long exons that are found in all amniotes. And I want to talk about gene tree discordance first. I don't think enough attention has been paid to things other than integration and incomplete lineage sorting, because there are plenty of other possibilities in terms of why gene trees might be discordant with each other. Then I want to present an example using our expanded relic data set that involve iguanas and chuckwallas in a paper that should be published very soon. So you can get a sense for what's happening there and also be able to compare different genomic subsets. And then finally, I've got a new explanation, I think, is why all of these relic loci, at least the majority of them, are sort of tied together. They all are dominated, or 80% of them are dominated by disordered domains, and I want to sort of talk about what that might mean in terms of modeling and understanding function. So I think that marker selection is something that we always have to think about, even we're getting these whole nice complete genomes, but we still have to, with whole genomes, we need to align sets of orthologous loci that are needed sort of for phylogenomics, and we need to make decisions. Do we want gene trees? Are we going to stick to SNPs for the genome? We're going to only look at conserved or variable loci? What sort of filters are we going to apply? There's lots of decisions that have to be made, and I feel like not enough attention is being paid to how those filters are decided upon and how they're applied. So again, I want to talk about gene tree discordance first. Very often the gene tree does not match the species tree, and I think I'll blame Wayne Madison for this because in 1997 when he sort of made popular, didn't come up with the idea, but he made popular the idea that gene trees don't match species trees, and for all of his discussion he assumed that the gene trees were accurate, right? He assumed that that all of the gene trees were accurate representations of the history of those genes, which is not true often. Right? And so if they are accurate, then incomplete lineage sorting and integration really are their only two options, or there's a couple of other options, but those are the two main options for why gene trees might differ from species tree. But there's other possibilities. One of them is systematic error or model inadequacy. So we saw a little bit of that, I think, in the last talk. And the best example I can think of are squamate mitochondrial genomes. You analyze those, no matter how you treat them a priori, you get a strong support for agamids and snakes being sister taxa to each other because of convergence, right? And it is not the gene tree, and it's not the species tree, right? It's another option. It's not representative, and it's because there are major shifts in function. So anytime you have major shifts in function for orthologs, you expect to get the wrong gene tree that might be very well supported. How often does this occur in the nuclear genome? We don't know, and we sort of hope that that answer is very low, right? But we don't really know. And then finally, gene tree estimation error, right? We're dealing with large data sets that are highly invariant. Most of those gene trees may correspond to just random sets of gene trees. They're not the gene tree, they're not the species tree, they're something else. So gene tree estimation error really has not been considered well enough. So when information is limited, right, you've got 2 informative characters and 100 taxa, and your gene tree is going to represent not the species tree and not the gene tree, it's going to be something else. And it's most likely to occur with short loci, slow evolutionary rates where you've got UCEs or maybe a set of short exons. And so you can come up with solutions. You can bin them together, you can concatenate them, you can filter the loci based on quality. So there's a number of ways you can deal with this, but I feel like it needs to be thought about more carefully. And when we look at whole genomes, whether we're talking about choosing from complete genomes or designing probe sets, we can decide we don't want gene trees and we can just use RAD-seq and get SNPs, or we can use exon capture. Ultra-conserved elements and anchored hybrid enrichment are the 2 most commonly used subsets that have been used with probe sets, but also being pulled out of whole genomes to be able to look at them. So this is the sort of gene subset that the bottom line is, this is the best subset and all of the other subsets are crappy. So that would be the take-home message here, that my genes are better than your genes. But the idea really is that the idea behind these rapidly evolving long exons is that they have to be at least 1,500 base pairs in length. I started with human genes are at least 1,800 base pairs, and then some of the squamate ones are a little smaller, so I took those. They have to be more rapidly evolving than RAG1, and that's just maybe that's an arbitrary decision, but it's at least it's a criterion. Most people don't have any criterion or how variable their loci have to be, so that's a decent one because RAG1 is used very often, and it's pretty good actually. It has to be present in all amniotes, and we derive this from the complete set of human exons. And most large human exons are in fact highly variable. And these also, all of them produce accurate gene trees. So Ben Karen came up with an orthology assembly and concatenation for being able to take the 287 loci that we have and be able to pull them out of existing genomes. So this is Ben's animation here. Because they're highly variable, you often get sort of fragmented blast hits, and so this is able to address that. And so If you want to get the scripts, they're available at Ben's GitHub, and it does a really good job. Especially the better the genome is, the better job and the easier it is to pull out all of the relic loci from completed genomes. So I want to talk about a paper that's going to be coming out very soon. I was hoping it would come out by the time that I gave this talk, but it hasn't yet. And this is iguanas rafted more than 8,000 kilometers from North America to Fiji, and that's not what this talk is about. It's the main point of this paper, but so my collaborators and most of the results that I'm going to be mentioning that are relevant to relic were completed by Ben Karen and Simon Scarpetta. So those are the two that really did all of the work that I'm about to talk about here. And we designed probes from 287 relic loci that totaled about 1 million bases. We sort of filtered the UCEs to find the best ones, 1,000 of them with the highest phylogenetic utility, and then included 500 base pairs of flanking region sequences. We had 316 anchored hybrid enrichment loci, 38 loci, which are from the squamate conserved loci probe set, and then all of the genes on the X chromosome. The X chromosome is fairly small, so there's 328 genes, and we looked at all 3,625 exons. And that in analyses that were by exon and then by gene, because in theory, the X chromosome should be evolving faster, and it should give you a nice data set, but it does not, in fact. So here's the main results from the paper, which are, again, biogeographic, and I'm not going to go into it, even though it's interesting. But the main point with respect to relic loci is that the inclusion of the relic loci are really what provides a well-supported sister taxon relationship between the California desert iguana, Diptosaurus dorsalis, and the Fijian iguana. So those are the top 2 taxa on the tree. Other phylogenomic data sets have come up with a different relationship, right? So there's been a UCE, I think it was a UCE data set that came up with Fijian iguanas, a sister to all of these other iguanas that are listed here, but this one, the relic, really sets it up, and it makes the timing much more compatible with the origin of Fiji itself. So, and we're also able, because it's a fairly small set of loci, we're able to do full multi-species coalescent analyses. We're able to throw in subsets of the other data sets that I mentioned as well to be able to analyze all of this. So without going into any of the interesting stuff that involves actual interesting lizards, let's talk about the markers themselves and comparing them. And again, Simon Scarpetta did all of these graphs. So first, if you look at the lower left graph there, that shows the average bootstrap support, right? And so what we see is that relic loci have very high bootstrap support. In fact, if we were to throw out a number of the poorly performing relic loci, they would all have bootstrap supports over 90% across the board for all of these. So the different colors are, in case you can't see it, the purple are the relic loci, and the green are the anchored hybrid enrichment, and then the 2 on the right are the X chromosome markers, and the yellow ones are the UCE markers, right? So, but this doesn't just have to do with it doesn't have to just do with support, right? And you can sort of see why the relic loci perform so well is that they're not only have more informative sites, per base pair they have more informative sites on average, right? So this is the proportion of informative sites on relic loci are extremely high. And so if we were to think, this isn't just like reducing support, it also involves the topology, and that's sort of the key in terms of gene tree concordance. So if you look, relic loci have high gene concordance factors and high similarity to the species tree, right? And so if you had just had a UCE data set from here, and you had a low gene tree concordance and you had high dissimilarity, you might say, well, there's lots of incomplete lineage sorting, and there's lots of integration, or you might come up with some story like that, but it's not true. You just have a bunch of crappy gene trees. So that's the bottom line. And it's true for the X chromosome as well. We kind of had high hopes that the X chromosome might do something interesting, but it doesn't. It comes out pretty much the same as the UCEs. So really the relic loci really shine in this context. This was true back in 2020 when we looked at all squamates, and you can see that there's exact species matches for where the relic loci exactly matched all squamates. A number of them do, and then if you look at a more closely related clade, colubroid snakes, over 80% of all the relic loci exactly match the species tree, right? And that's not true for the other markers, and it's not that you have some special species tree set of markers. No, you just have well-defined markers, and that the reason that the other markers don't match the species tree is because they're not very good. That would be the bottom line for that. So why is this? I wanted to give myself enough time to discuss this sort of new information. So we knew this before back in 2020 that if we look at all of the relic loci, we see strong signatures of relaxed purifying selection. So that's not terribly surprising. And we look at UCEs, they're undergoing high purifying selection, so it's 3 times higher than the protein coding regions on average, right? So that's pretty clear, but you sort of left with this lingering question, is there anything that ties them together, or why do big exons tend to have relaxed purifying selection? Because that's really generally true. Also, because these are all protein coding, we can really model nucleotide evolution, strength of selection, amino acid evolution, and so we can do a lot of different things that we can't do with any other genomic subsets. So here's what's similar. 80% of the relic loci have large intrinsically disordered domains. That is, these disordered domains. And what's a disordered domain? It's a domain that lacks a three-dimensional structure. So I can't show you any pretty pictures of them because there aren't. There's no pictures of the structures of them because they'd have. And it's not just a leftover bin. It's not that any segment that doesn't fit into one of the functional categories is disordered. These are a set of amino acids that define them as being disordered domains. And this would not be discovered through traditional functional analyses because these occur in a variety of genes that do a wide variety of different things. And so was able to use interpro scan to be able to find all these disordered domains in a wide variety of genes with a bunch of different functions. So what are those functions? I'll give you 3 examples. So often they are spacers, and this is the ideal situation where you've got, example is POLQ. It's a single gene that contains both DNA polymerase and a helicase, right, which are highly constrained on either side, and in the middle there's this huge disordered domain, which ensures that only long pieces of DNA get repaired. And so experimentally they've been able to cut down the disordered domain, and it cuts shorter and shorter pieces of DNA. So you've got this ideal situation where the length is constrained, but the base pair composition the amino acid composition isn't very constrained at all. There's an overrepresentation of relic loci that are transcription factors or DNA repair enzymes or histone modifiers that are binding to other proteins or DNA. And we know that the disordered domains from studies of them involve alternative configurations that allow them to bind to a variety of markers. And finally, enamelin is a good example of something that involves structural spacing, right? So we're able to, there's a gigantic, again, disordered domain that ensures that enamelin is laid down in a matrix in a way that sort of structurally spaces them out in the right way. Again, we've got spacing's important, but the constitution of the amino acids isn't, and so you get this great marker like that. So, in conclusion, relic loci are a subset of the genome that produce highly informative gene trees that tend to match the species tree better than any other markers, and that's because the other markers contain a large proportion of gene trees that are inadequately resolved. You can analyze them with amino acids or codon-based modeling, and I think that gene tree discordance deserves more attention than it has received, and disordered domain seems like an exciting, interesting thing to be able to look at more carefully in these relic loci. Thank you.
So, hi everyone, my name is Rémi Allio, and thank you for being. Today I'm very glad to present you the work of Benjamin Zalveda, who is currently doing his PhD in our lab in Montpellier. And he's not attending this conference, but he will be at the International Conference of Entomology next month in Japan. So if you want to meet him, you can meet him there if you're going. So in the present study, we aim to evaluate whether the interaction between plants could provide ecological opportunities for insect host plant shift. So to begin this presentation, I would like to introduce you quickly why we are, as evolutionary biologists, interested in plant-insect interaction in the first place. So when we look at the relative global biodiversity of extant animal groups, we see that there are more insect species on Earth than all other animal groups combined. In fact, today, the insect biodiversity counts more than 1.2 million described species, and we know that there is much more to discover and describe. And most interestingly, half of this biodiversity is attributed to herbivorous insects. And this is not that surprising when we look at the diversity of plants, which is also enormous. And the evolution of this extreme biodiversity in both groups is often attributed to their interaction. And when we look at the evolution of the proportion of herbivorous insects through time, here in beetles, for example, we can see that the insects interacting with plants show bigger rate of diversification with more and more herbivorous insects through time. But one can ask, what is the role of this interaction in their reciprocal diversification. And this question has led to several theories, and the most well-known theory is called the escape and radiate hypothesis. And proposed in 1964, this hypothesis suggests that when a plant develops a defense against an insect, it increases its fitness, and then with time leads to the plant's diversification. After that, insects can adapt to these new defenses and begin to feed on this newly diversified group of plants. This is what we call an plant shift, and it leads to it provides insects with new niche, and then they can diversify in turn. So even though the escape and radiate hypothesis is not the only hypothesis to explain that, all the theories agree at least on 2 points. Host plant shifts lead to new ecological opportunities and lead also to reproductive barriers between species and then to speciation events. Altogether, these processes contribute to the diversification of both insects and plants. So the remaining question is how these shifts occur in the first place. In other words, what are the ecological opportunities leading to host plant shifts in insects? And to explore this question, in the present study, we look at the evolution of a Smicrolechini, which is a tribe worldwide, which is very diversified and found worldwide. But they are also presenting an unexpected houseplant repertoire at the tribe level. Indeed, while most insect genera or tribes feed only on a single houseplant family, these weavers feed on 4 distinct plant families. And on top of that, the Smicrolechini are among the very few insects known to feed on holoparasitic plants. So, and in some cases, they are forming gall, as you can see in the picture. So holoparasitic plants are parasitic plants that depends on their host plants to fix carbon and develop and survive. But they also feed on hemiparasitic plants, which are parasitic plants that need their host, but they remain photosynthetic in some degrees. And finally, they also feed on non-parasitic plants. So in total, it is 4 distinct plant families, ranging from parasitic plants to non-parasitic plants. And some families have several levels of parasitism. For example, the Convolvulaceae present both holoparasitic plants and non-parasitic plants. And the Robranchaceae present 2 levels of parasitism. So the question is, in this study was how the different host plant shifts occurred in the evolution of the Smicrolechini. And one of the possibilities is that the interaction between the plants themselves had an impact in the host plant shift dynamics of this insect by providing the ecological opportunity for the shift. Indeed, some of the current weevil species are, some of the current host plants of weevils are interacting between each other, with the Convolvulaceae, for example, being known to parasitize the 2 other plant families used as host plants for weevils. So to test this hypothesis, we used a phylogenomic approach, and if this interaction played a role in the host plant shift dynamic in these weevils, we expect that the common ancestor of all Smicrolechini fed on a parasitic plant, and that the shift toward the non-parasitic plant occurred only in a second time, as opposed to the alternative hypothesis where the parasitic plants were colonized only in a second time. So to test this hypothesis, and because we know nothing about the evolution of Smicrolechini, we reconstruct the first phylogeny for this tribe using a phylogenomic approach and a anchored hybrid element enrichment. Sorry about that. And I will quickly go through the method, but we can discuss that further after my talk if you want. So for this study, we sampled about 50 species over 10 years of sampling and exchange with museum. Due to their worldwide diversity, it is kind of hard to find them, and we don't have all of them. And even though our sampling was incomplete for the tribe, we were able to sample all the ecotype of the tribes, which is what we need for the question. So then we targeted more than 900 loci that were supposed to be considered at the coleoptera level. And after sequencing, each locus was assembled and using an iterative assembly approach, and we used a custom pipeline to isolate and filter all loci. So as suggested in the previous talk, we were not really happy with this set of probe, and we are currently designing a new set of probe for Weevils, and we got 2 funding for that, so stay tuned, there are definitely more coming in Weevil phylogeny field. So then, anyway, after that, all loci were combined in the supermatrix, and we used IQ3 to infer the phylogeny of the tribe. The best fitting model was inferred with partition filer, and some species for which we had only mitochondrial COX-1 sequence were grafted using COX-1 sequences to improve sampling. So here is the first phylogeny obtained for this group. So without going further into detail, we can see that the phylogeny is robust with only 1 node with low support, and the Smicronychini are monophyletic. However, the genus Smicronix, which include most of the species of the tribe, is not full monophyletic, with 1 genus found in the middle. So we need to review the taxonomy of the group. But what about the host plant shift dynamic of this tribe? So based on the current host plant use of our Weevil, we performed an ancestral stage reconstruction to infer the host plant shift event in the evolution of Smicronychini. And what we found is very interesting. So the ancestral host plant was indeed a convolvulacea. So we are not sure if it was a parasitic plant or a not parasitic plant. But then we can see that species feeding on a parasitic plant diversified. So in orange, you can see several lineage of weevils feeding on convolvulacea, parasitic convolvulacea. And then we can see several shifts toward non-parasitic plants, but also towards other parasitic plants. So this is like a summary of what we observed. So we observed 3 types of shifts, always from parasitic plants toward other plant families. And in the first case, it seems that all the barriers to host plant shift opportunities, such as the need to be at the same place at the same moment, the ability to detect your host plant, or the ability to deal with plant chemical compounds, were bypassed due to the interaction between the plants themselves. And so in other words, this interaction between plants provided the ecological opportunity for the insect to adapt to another plant family. And then we have a very interesting case with Smicronix quadrifer. In this species, the female lay on the parasitic plant, and then the larvae migrate from the parasitic plant to the non-parasitic plant during its development. So basically, this species is growing in 2 plant families due to their interaction. So unfortunately, we don't have this species in our dataset, so we try to contact museum without any success. So if you know an entomologist living close to Tucson or Phoenix in the US, please reach out so we can try to find this species. I don't know about you, but I really want to know where it falls in our phylogeny. Regarding the shift toward other plants, parasitic plants, we know that we can find them at the same place at the same moment, and we can imagine that all these parasitic plants could share some host plant and thus can be in contact in a way. It might be enough to create the opportunity for the host plant shift. So the take-home message here is that the interaction between parasitic plant and the host plant make up some sort of facilitating bridge that can be crossed by insect toward new potential hosts. And so we have several perspectives on this work. So we want to make a dated phylogeny and diversification analysis associated with parasitic plant association to see if this particular host plant shift dynamic in this tribe is indeed associated with birth of diversification. We also want to review the taxonomy of Smicronychini, and we want to go further on the mechanisms involved in this kind of shift with parasitic plants and non-parasitic plants involved. So maybe if we can find some Smicronix quadrifer and go further on this case. So to finish, I would like to thank all my collaborators, and the first version of the study is available on the archive if you want more information, and thank you for your attention.
Hello. My name is Eric Hagen. I am a postdoctoral fellow at the University of Toronto, where I work with Tomo Parents Fukuchi. And for the past few months, we've been collaborating with some friends of ours, including James Pease at Ohio State University, as well as Stephen Smith at the University of Michigan, to investigate the relationship between phylogenomic discordance and the emergence of morphological novelty. And we're thinking about a lot of different ideas, how to answer various questions of ours. So this is going to be more of a conversation about those ideas and questions than about some hard results. But I hope to talk more about those perhaps next time. So most of you probably are aware, but in case you are not, individual gene trees can disagree with other individual gene trees and also with the species tree. And this is called discordance. I did my PhD working with methods that take the species tree as a given. And when I read about this, read Wayne Madison, it really caused me great anguish at first, I gotta say. But discordance is ubiquitous, and these sorts of conflicts are a great source of information about various different things and can be very informative for uniting population processes with macroevolutionary patterns. So some of the causes of discordance include things like whole genome multiplication. That's something that I think about a lot and we'll talk about a little bit later. Things like incomplete lineage sorting and single gene duplication and loss, as well as selective mechanisms like rapid selection or hybridization. And discordance can also result non-biologically from data containing errors or insufficient information. So for another example, these are two gene trees taken at random from our dataset on Papionin Old World monkeys. The one you see here is Runguicebus kapungi, a very recently described species that is believed to be a hybrid between Papiocynocephalus and Lophocebus. In this clade, there is a lot of hybridization, and because there is some information about the processes going on within this clade, we're using it to sort of calibrate our analyses before we move into our analyses of large clades of flowering plants. But as you can see, these topologies are different from each other, and they're also conflicting with the species tree. So to get to the point of what we are looking into, Tomo, who I work with at the University of Toronto, has done a lot of work, including in this paper, correlating phylogenomic discordance with morphological innovation in deep time. This is something that we see in many different clades, from plants to animals. And of course, discordance is not a mechanism for innovation itself, but we have to look at where we find these discordances that coincide with innovation and ask ourselves what specific mechanisms might be causing this among all possibilities. So like I said before, during my PhD, I studied polyploidy, particularly its relationship to diversification using things like SSE models. I'll talk very briefly about this research that I'll plug. If you happen to see me present on this at Botany last year, I'm sorry that this is not already out. It will be out very soon, I swear. But this was trying to extend traditional HISI models that account for hidden variation to say, well, in the polyploidy world, we often don't see a transition to polyploidy and then immediately a diversification shift. There are often lags. And in this model, we are incorporating the ability to have lags. But the point for this talk is that in this and in many of my other projects that I worked on during my PhD, I didn't find much evidence to support the idea that polyploidy leads to increased diversification regularly across many different plant clades. Of course, some of you in the room may say, well, that's bananas. You don't know what you're talking about. And that's okay. That might be true. But because I haven't seen much evidence, from my own work at least, that there is a relationship between polyploidy and diversification, I had been thinking, well, maybe whole genome multiplications aren't causing these influxes of innovation as opposed to other processes that could be creating it. But recently I've been thinking about the fact that maybe we can have innovation without increases in diversification. This is something that was pointed out by Dan Hrabowski in a paper from 2017, in which he argued that comparative biologists often confuse key innovation in adaptive radiation with just simple increased diversification. We have to keep in mind that key innovations can lead to increased clade diversity in situations where they don't also increase species richness or the diversification rate. In this case, we have a clade labeled in blue, which develops a key innovation and reaches its carrying capacity at the tree at T2, time 2. And then at time 3, the key innovation clade and the original one are monophyletic due to turnover, such that the overall clade is, in fact, more diverse, but the diversification rate in the original clade has remained unchanged. So these are things that we've been thinking about. How do we specifically plan to assess innovation over time? Well, we're working with our colleagues at the University of Michigan, particularly Stephen Smith, Selena Smith, and Stephen Smith's PhD student, Will Weaver, who have developed a very fascinating machine learning tool called Leaf Machine. Specifically, they're now on Leaf Machine 2. And what it is able to do is identify herbarium specimens efficiently. And on top of that, it can also automatically capture various leaf trait measurements with computer vision techniques. What we hope to do is to use this tool to capture a very large trait data set, including both present-day herbarium specimens and fossil specimens. And our goal is to identify periods of major evolutionary innovation in flowering plants based on these leaf measurements and determine whether these periods coincide with elevated phylogenomic discordance, and potentially as well infer the population processes underlying these patterns. That's the goal, at least. So now on to some questions that we're thinking about in particular. These two graphs also come from a recent paper by Tomo. But we were asking, does the variation displayed by taxa that happen to be hyperdiverse originate from ancestral lineages that were very polymorphic? So this paper was spurred by the fact that there is a lack of research on the consequences of the long-term maintenance of phenotypic variation. And what these graphs specifically show is that polymorphism is polymorphism maintained by negative frequency dependent selection. In scenario A on the left, we have budding speciation that causes a bottleneck resulting in random fixation, whereas in scenario B on the right depicts fixation due to positive selection. Part of this paper focused on crinoids, and it found that descendant lineages of long-lived, highly polymorphic ancestors tend to be less variable than their progenitors. And what this paper touches on that I find very interesting is the possibility that peripherally isolated crinoid populations may arise from single polymorphic long-lived ancestors. And in this case, drift may be very strong and caused random fixation such that only those populations with stochastically fixed beneficial alleles may persist and leave descendant lineages. And this leaves room for the possibility that species selection, selection at the level of species rather than the population, differentially culls descendant lineages on the basis of the randomly fixated alleles that they inherit. The takeaway is these are the sorts of what at least I find fascinating evolutionary processes that we think our methods may be able to uncover. So one thing that we're thinking about is how do we expand models of ancestral polymorphism from binary states, such as in Abba-Baba, to multiple states or even further to continuous states, quantitative traits. The distribution would have to be pretty bimodal in a continuous trait to constitute a polymorphism. Otherwise it's just continuous variation. And on top of that, but what we're thinking about is modeling discordance of quantitative traits could involve things like shifting of distributions, expanding and contracting variance, etc. A paper that came out recently that sort of does this, something that we're thinking about a lot, is one by Feng et al. From a couple years ago. They studied discordance in Australian and South American marsupials, focusing on the monotypic clade containing the South American Monito del Monte, which shares many anatomical traits with Australian marsupials and shares a very similar genome with the Australian marsupials, specifically in the clade Diprotodontia, which contains koalas. Incomplete lineage sorting likely contributed to the hemiplasia here in morphological traits. And we can hope that, as in this case, the morphological hemiplasia that we may observe will map very easily onto genomic discordance, but we know that that may not always be the case, depending on what we're studying. One final question I want to discuss here that we're considering is whether genetic integration can prevent rapid evolution even when processes we infer at the population level would anticipate it. This could explain the fact that discordance and innovation are not universally coincident. And it has been suggested that shifts in the structure of integration can explain major morphological changes over time. So right now we're exploring ways to model shifts in evolvability of morphological characters over time. For all these things, including this, if you have ideas about these things or considerations of your own, I would definitely love to talk to you about that. One final thing I want to touch on is at the beginning of this talk I said that we were going to be correlating discordance with morphological novelty. And this entire time I've been telling you about innovation, which very recently I was looking at the literature and found out that there's a lot of discourse about how these are two different things. So I'll talk about that briefly. For example, Günter Wagner in 2015 defined novelty as specifically the evolution of new structures, whereas innovations, according to him, are the evolution of new functional capabilities. So I'm a plant person, so as an example, novelty would be the evolution of flower structure, whereas an innovation might be something like taking the simple dichotomous branching in things like ferns, and then they evolve into the more complex networks that we see in angiosperms that allow for higher photosynthetic rate. Is discordance related to these things in different ways? I don't know. See me after the talk. I'd love to talk to you about that. But yeah, my main takeaway for you is I'm excited about this research, and I hope to tell you more about it in the future. And I'm very happy to be working with this great group of people, particularly Tomo and James Pease, and the excellent students in the Parents Ficucci lab, especially Michelle and Thomas. So thank you very much for listening. Do we have time for questions? Yes. Thank you.
Have you thought at all about like incorporating sort of potentially like ancestral range occupation or kind of like the social component of
So, yes, we have. So we're working with this baboon clade first, and we have done some mapping of ranges trying to look at climate data specifically, but right now we haven't delved too deeply into it, so I would love to talk to you about that if you have ideas. Thank you very much.